Hi everyone. This is the third lecture for your chapter Respiration in Higher Plants. So, in the previous class, we discussed the very vital and very crucial pathway which is seen in all living organism, which is none other than glycolysis. Okay, we also saw that it is anaerobic process, which means it does not require oxygen and it also does not release any carbon dioxide. We saw the 10 steps which are required for glycolysis, and in the end we also saw the tally, okay, how many ATP are produced and how many NADH are produced. Today we will be discussing the fate of pyruvate. Okay, what happens to the pyruvate that is made at the end of glycolysis. So if there is presence of oxygen, so there is some oxygen present, then the cell will go for cellular respiration, okay, aerobic cellular respiration. Aerobic, anywhere where you read, it means in presence of oxygen, fine. So in presence of oxygen, pyruvate is converted into acetyl CoA, and this acetyl CoA goes to Krebs cycle. From Krebs cycle, the reducing powers, that is NADH and FADH2, will then go for electron transport chain and will give you ATP. The carbon from pyruvate or from the glucose, they will be released in form of carbon dioxide. So this here will be the complete oxidation of glucose. On the other hand, if the conditions still remain without oxygen, okay, then without oxygen, the next reactions that occur are called fermentation, okay, and we shall see just in a minute why this fermentation is necessary, why we cannot leave it at pyruvate, okay. So fermentation could again be of two types. In yeast you see alcoholic fermentation, that is it makes alcohol, okay, from pyruvate. And then the second type is called lactic acid fermentation, in which it makes lactic acid, okay, mostly your muscles, they make lactic acid in absence of oxygen. Let us look at this one by one. We start with ethanol fermentation, okay. So ethanol, it is produced by the yeast. Any other type of bacteria or any other type of living organism will produce other organic acids, okay. Just as an example, the lactobacillus bacteria in your curd will produce lactic acid. Same way, your muscles will produce lactic acid. Then there are some bacteria which may even produce other organic acids like malic acid or oxalic acid or some other acids, okay. So this is just an example. The two main fermentations that take place are ethanol and lactic acid. Ethanol is made mostly by yeast. So it occurs in yeast when incomplete oxidation of glucose is achieved under anaerobic conditions. So remember, just focus on this part, okay? Remember during glycolysis, so this is the point, okay? From glucose, we converted glucose into two molecules of pyruvic acid or pyruvate. During this whole process, we were making net 2 ATPs, okay? Net meaning, like how I told you last time, net profit, right? So net 2 ATPs were made, and using NAD plus, 2 NADH were made. Is that much clear to you? Now what happens is, for this pathway to keep going on, okay, for glycolysis to keep running, you require free NAD plus, right? If you do not have NAD plus, then you cannot make NADH. And if you cannot have NAD plus, then who will you donate the hydrogen to? Okay, remember the step where the phosphoglyceraldehyde gets converted into phosphoglycerate. Clear? So during this process, you require a hydrogen acceptor, electron acceptor, which is nothing but your NAD plus. Okay, only if this NAD plus is present, the pathway will move in the forward direction and give you to pyruvate. Now, if the cell keeps on running in anaerobic condition, okay, so anaerobic condition meaning there is still no longer any oxygen which is present. In which case, what will happen after some time? This NAD plus will get over, right? You do not have any more free NAD plus. So if NAD plus is not there, how will the glycolysis go ahead? It cannot go ahead. Okay, so nobody is there to take the hydrogen and hydrogen and electron. So to make sure that glycolysis does not stop, as you can see, because glycolysis is the point where you are producing two ATPs. to make sure that you do not stop getting energy. This fermentation was developed. Okay, so now from this 2-pyruvate, there is an enzyme called pyruvic acid decarboxylase. Okay, so pyruvate decarboxylase or pyruvic acid decarboxylase, both of them are same. Pyruvate decarboxylase, as the name suggests, D means to remove and carboxylase means to remove carbon dioxide. So this enzyme, it removes carbon dioxide from pyruvate, and pyruvate is a 3-carbon molecule. So if one carbon dioxide leaves, it gets converted into acetaldehyde. Here, now acetaldehyde is converted into alcohol or ethanol with help of another enzyme called alcohol dehydrogenase. Clear? So alcohol dehydrogenase does what? It converts acetaldehyde to ethanol. Now remember, I had told you can convert aldehyde into ethanol if you reduce it. Okay, on reduction of aldehyde you get ethanol. So for this reduction, what will be used? You require some protons, or hydrogen, right? Or you require electrons. Where do they come from? They come from the NADH. So here the NADH will donate its proton H plus, it will donate its electron E minus, and so the ethanol will be formed. Okay, acetaldehyde will be reduced and ethanol will be produced. Clear? Once it donates its H plus and electrons, the NADH becomes NAD plus. And now this NAD plus is free. Okay, it is available for the glycolysis reactions. Clear? So this is how it is the cell. It makes sure that NAD plus is never less. Okay, so that the glycolysis does not stop. If NAD plus levels become low, okay, they go down, then glycolysis will become slower. So to make sure that NAD plus levels are maintained, this NADH, okay, is used to make ethanol. Remember, during this process, during alcohol fermentation, you get carbon dioxide. Okay, so this is why in fermentation industry, where they make liquors and alcohols and beers, you must have seen that beer is a alcoholic drink which has carbon dioxide in it. Okay, it shows bubbles in it. So these bubbles are nothing but they are carbon dioxide gas. Clear? Okay, so glucose to pyruvate, pyruvate to acetaldehyde, and acetaldehyde to ethanol. This is ethanol fermentation. Clear? Now we come to the lactic acid fermentation. This lactic acid fermentation, it occurs in bacteria. It occurs in some parasites, endoparasites specifically. Endoparasites are the parasite which live inside your body. Okay, so they will be living in some tissue, in some organs, or even in your GIT, in which case they are not getting enough oxygen. So they undergo lactic acid fermentation. When you are running, let us say a marathon, okay, or you have to run a 400-meter sprint, then what happens is your muscle, okay, it is not getting enough oxygen, so it will start doing anaerobic glycolysis, okay, or anaerobic fermentation or lactic acid fermentation, right? So from glycolysis it will receive the 2 ATPs and it will convert the pyruvate, which is the end product of glycolysis, into lactic acid. Here, so here are the reactions. It is a one-step reaction, okay? Unlike the ethanol fermentation, this reaction happens in one step. There is only one enzyme required, which is called lactate dehydrogenase. Clear? So what happens now? Here also you have this 2 NAD plus, which are reduced, okay, and they become NADH during glycolysis. Now, to make sure that the glycolysis keeps running, you require more and more NAD plus. To remove this H plus and electron from NAD, this is why the lactic acid fermentation happens. Okay, so 2 pyruvic acid molecules with help of lactate dehydrogenase enzyme, they will be reduced and give you lactate. Okay, so from pyruvate you get lactate, and NADH is utilized during this process to reduce the pyruvate molecule and give you lactate or lactic acid. Clear till here? So you get free NAD plus to make sure that glycolysis keeps continuing, and you make sure that pyruvate is getting converted into lactate. So this is the whole process of lactic acid fermentation. Clear till here? Right. So after understanding glycolysis and what is this anaerobic respiration or fermentation, right? Let us find out what is the efficiency of this anaerobic respiration. Clear? I had told you in the first class itself that we will later on find out this efficiency, and you will be surprised to find that even um, after all of this effort that is put, okay, by the cell, the efficiency is not very much. Chalo, let us begin. Take out your notebooks and um, uh, solve with me, okay? Yeah, by the way, on this right hand side, this is the bomb calorimeter, okay? And bomb calorimeters are used to find out the calorific values of different food items, okay? So if you look at any packets of snacks or of any edible things, okay? So behind the packet, they will write down energy values or nutrition values in which calories are mentioned. That energy you get from this, let us say Coca-Cola, is 500 calories or 200 calories, right? So how do they find out these calories? They find out by doing this, okay? They burn the substance, and that energy, that heat energy which is generated by combustion, is used and it is measured. The temperature change is measured, okay? And then they find out that these many calories are produced by 1 gram of let us say Coca-Cola, or 1 gram of potato chips. Clear? So that is just a very brief way of telling you that how they find out the calorific values. Chalo. So theoretical energy yield from burning 1 mole of glucose. Fine, 1 mole of glucose if you burn in bomb calorimeter, you get minus 686 kilocalories per mole. Okay, so this is the delta G value actually, but let us just understand that this is the calorific value of glucose. If you burn 1 mole of glucose, you get minus 686 kilocalories. Clear? Why is this minus? This minus is showing that energy is getting released, okay? So it is an exergonic process. Exergonic process meaning heat energy and other sorts of energy are getting um, the reaction is giving out energy, fine? So minus 686 kilocalorie per mole of glucose. This is theoretical value. Now ADP and the third phosphate, okay? So this is just another way of writing ATP, okay? So basically you have adenosine, and with adenosine you have 3 phosphate groups attached, right? So this ADP and the third phosphate, between them there is this bond, which is nothing but your phosphate bond, anhydride bond. So this phosphoanhydride bond, when it is broken, it releases minus 7.6 kilocalories per mole. So 1 mole of these bonds broken will give you minus 7.6 kilocalories. Clear till here? So when ATP is broken, you get minus 7.6 kilocalorie. When 1 mole of glucose is used, it gives you minus 686 kilocalorie. Now anaerobic respiration of 1 mole of glucose is giving you 2 moles of ATP. This much is clear to you. We have just seen that net ATP gain from glycolysis is 2 ATP. Clear? So 2 means 2 moles when you write down in a reaction. So 1 mole of glucose is giving you 2 moles of ATP. So that means 2 moles of ATP. So 2 multiplied by how much you are getting energy per mole of ATP, you are getting minus 7.6. So 2 into minus 7.6. When you calculate the value, you get minus 15.2. Okay, so you're getting minus 15.2 kilocalorie per mole of glucose. Are you able to understand this calculation? So we found out glucose. Okay, glucose is 686. ATP is 7.6. Now we know that 1 glucose is giving you 2 ATP, right? And we also know the calorific value of ATP, so it is 7.6. So 2 into 7.6. Clear? So this is 15.2. So from 1 glucose, you are getting only 15.2 kilocalories. Are you able to understand? From 1 mole of glucose, 15.2 kilocalories. Now to find out efficiency, how efficient is this process? What do? You divide this, okay? You divide 15.2 by the theoretical value, the actual value. If you oxidize glucose in presence of oxygen, okay, completely oxidize it, you get minus 686 kilocalorie. Here you are getting minus 5.2. So the percentage efficiency, if you want to find out, it is minus 15.2 divided by 686 and multiplied by 100, which comes to 2.2. So this is how efficient anaerobic respiration is. If in anaerobic respiration, okay, which is respiration without the presence of oxygen, when you partially oxidize glucose, you can get only 2% of the energy stored inside the glucose molecule. Okay, that is what is shown by this number. Fine. If you are having some problem understanding this, just pause this slide, okay, and try and find out what this is meaning, okay, how this calculation is made. If you understood the whole process, very good. If you have not understood, just pause for a minute and reflect upon it, okay. You will be able to understand what calculations we have made to find out the percentage efficiency of anaerobic respiration. This is just to show you that even after all of these reactions, 10 reactions of glycolysis, 2 of alcohol fermentation, or 1 of lactic acid fermentation, you are just receiving 2% of the energy stored inside a glucose molecule. Now let us talk about metabolism in presence of oxygen. What happens if oxygen is present? What is the fate of pyruvate? So this is called aerobic respiration. Aerobic respiration is complete oxidation of the organic compound, okay, of any respiratory substrate. As we had discussed, it could be carbohydrate, it could be fat, it could be protein, or even some organic acid. So any organic compound, when it is completely oxidized, okay, in presence of oxygen, and in the end the product you are getting is carbon dioxide, water, and a large amount of ATP, then it is called aerobic respiration. This type of respiration is most common in higher organism. Aerobic respiration, it occurs in mitochondria. Okay, so you require a mitochondria for aerobic respiration to take place. Clear? Even in bacteria, there is aerobic respiration. They also have Krebs cycle, but we shall not go into the detail. Just know that because bacteria, they do not have mitochondria, so for them, the Krebs cycle and all of those things will occur on its cell membrane. Okay, fine. So aerobic respiration, it occurs in mitochondria. Excuse me. So there are two main events, okay, two main parts of what happens in the mitochondria. So in the matrix of mitochondria, so here the dark blue part that you are able to see is the matrix, okay, it is the ground substance of mitochondria. It is nothing, it is fluid. Along with fluid, it has a lot of protein, it has this mitochondrial DNA, it has ribosomes, okay. So that this dark blue colored fluid which is shown here in the diagram is your matrix. So in the matrix, complete oxidation of pyruvate occurs, giving you 3 molecules of carbon dioxide. Okay, so that is the one part of reaction, one part of aerobic respiration occurring in the matrix, okay, mitochondrial matrix. The second step happens on the inner membrane. I hope you remember the structure of mitochondria. You have an outer membrane and then you have this inner membrane. So on this inner membrane, there are a series of reactions which take place. So there is passing of electrons. Okay, so the electrons, they are passed along different electron acceptors and electron transporters. And finally, the H plus, okay, so protons plus oxygen. Um, Protons plus oxygen and these passed electrons together, they will form your water, okay, H2O. And that is how the whole process will take place, and it will give you ATP also. So we will see this two parts. In matrix, what I'm talking about is called nothing but your Krebs cycle. So your Krebs cycle, it takes place in the matrix, and in the inner membrane, okay, in the mitochondrial inner membrane, there is electron transport chain which is coupled with the oxidative phosphorylation. Okay, so that happens on the inner membrane. So this is the overview. You have in cytosol, which is your cytoplasm, glucose getting converted into 2 molecules of pyruvic acid and 2 ATP you require. Okay, you get 2 ATP you require, 4 ATP you get, so net there are 2 ATPs. Then the high energy electrons, which is in form of NADH, okay, reduced power. So that NADH will be carried into the mitochondria. Same with pyruvic acid. Pyruvic acid will enter the mitochondria. It will undergo Krebs cycle. Krebs cycle will also give you direct ATP. We shall see that. And it will give you reducing power as well, which is in form of NADH and FADH2. Now this NADH and FADH will be carried to the electron transport chain on the inner membrane, and after electron transport there is oxidative phosphorylation, which will give you ATP in huge amount. Let us see it one by one. So by now we have 2 molecules of pyruvate. This pyruvate is still in the cytoplasm. What do we need to do? We have to perform the next reactions inside the mitochondria. So there is a linking reaction. Linking reaction, what does this link reaction do? It connects glycolysis with the Krebs cycle. Okay, so it is a link between the two. This link reaction occurs with help of an enzyme called pyruvate dehydrogenase. Okay, so this is your pyruvate, which is present in the cytoplasm. There is a transport protein on the mitochondria which will transport this pyruvate inside the mitochondria. Now, inside the mitochondria, pyruvate is first decarboxylated. So there is one carbon dioxide removed from the pyruvate. This carbon dioxide, as shown, okay, as highlighted here, comes from the carboxylic acid group. You can see COO. So this will form CO2. This CO2 will be removed. Okay. So first carbon dioxide is released here. Now the rest, okay, the remaining part of the pyruvate, which is these 2 carbons, they are oxidized. Okay, so from there the hydrogen is removed. Can you see NAD plus will get converted into NADH, which means electrons and hydrogens were removed from the pyruvate. Clear? So there is oxidation of pyruvate. Now because this carbon dioxide was removed, okay, this carboxylic acid group in form of carbon dioxide was removed. Here you this carbon has one more free valency, right? This carbon has one valency. To this valency there is a what you can say coenzyme. Okay, it is called coenzyme A. Remember biomolecules, okay, in the end we had discussed cofactors, and in that cofactors we also saw coenzyme. So coenzyme A, which is made from pantothenic acid, okay, vitamin B. So coenzyme A will be added. This is written just as CoA. So next time I tell you coenzyme A, or CoA, it just means coenzyme A. So this here coenzyme A is a long structure, and in the end it has this SH, which is sulfhydryl group. Clear? I hope you remember this SH group from the amino acid, right? So you write it like this: CoA, which means coenzyme A, SH. What will happen is this hydrogen will be removed, and this sulfur, okay, it will form a bond with this carbon. So you get this molecule which is called acetyl CoA. Acetyl because it has this C double bond O, so carbonyl group, and a CH3 group, and there are 2 carbons, so 1 and 2, so acetyl CoA. This is the coenzyme A. Clear? Fine. So just going through it all once again. Pyruvate enters the matrix, mitochondrial matrix, and undergoes oxidative decarboxylation. Okay, oxidative why? Because it is oxidized. Okay, hydrogens are removed from it. So oxidative decarboxylation, carbon dioxide is also removed from it. Catalyzed by pyruvate dehydrogenase complex to give acetyl CoA. The process gives you product which is acetyl CoA. Pyruvate dehydrogenase requires several coenzymes. Okay, it requires several coenzymes The only important ones I have mentioned are NAD plus, because we require NAD plus to accept the electrons and proton, and coenzyme A to attach to the acetyl group. The acetyl CoA then enters the Krebs cycle. Coming to the main part of the aerobic respiration inside mitochondria, it is called tricarboxylic acid cycle or Krebs cycle. It is also known as citric acid cycle because the first intermediate or the first molecule to form in the cycle is citric acid. Okay, so tricarboxylic acid cycle, citric acid cycle, Krebs cycle, all of these are names for the same thing. Krebs cycle is named after the scientist Hans Krebs, who elucidated the pathway. Like how Kelvin, scientist Melvin Kelvin, he found out the Kelvin cycle. Then Embedon Meyerhoff Parnas found out the EMP pathway or glycolysis. Same way, Hans Krebs found out the Krebs cycle. Okay, so that is how it is named. Let us start with the reactions itself. Okay, these are very simple. Just follow along. Fine. The cycle starts with an enzyme called citrate synthase. Citrate synthase does what? So, inside the mitochondrial matrix, you have acetyl CoA, okay, which was formed from the pyruvate, and oxaloacetate. Oxaloacetate we have again heard in the C4 pathway. Okay, it is the first molecule formed in the C4 pathway. So here you have oxaloacetate and acetyl CoA. As you can see, oxaloacetate has 4 carbons, acetyl CoA has 2 carbons. Both of them get fused, okay, they get connected, joined. This occurs with help of an enzyme called citrate synthase. So citrate synthase catalyzed condensation. Condensation is addition of water. So there is one H2O molecule added to the reaction of acetyl CoA with oxaloacetate and water yields citric acid. Okay, so what is the outcome? Input is oxaloacetate plus acetyl CoA plus water, and outcome or output is citric acid. So here you have 3 carboxylic acid groups, if you notice, okay, COO minus, COO minus, and COO minus. Because of these 3 carboxylic acid groups, this cycle is also known as tricarboxylic acid cycle, TCA. And because the first molecule to be formed is citrate, it is also called citric acid cycle. Clear? Now in this process, the coenzyme A from acetyl CoA is removed. Okay, so you get CoA or coenzyme A in its original form. Clear? So this is the whole process. Oxaloacetate plus acetyl CoA gives you citrate. The second step is where citrate is isomerized. Okay, so there is isomerism. So from citrate it is converted into isocitrate, and the enzyme for this is called aconitase. Okay, so this here is citrate, and the citrate is converted into isocitrate. Clear? Third enzyme is isosocitrate dehydrogenase. So we have seen till here OAA plus acetyl CoA gives you citrate, citrate gives you isocitrate, isocitrate, okay, is acted upon by an enzyme called isocitrate dehydrogenase. Okay, isocitrate dehydrogenase. Wherever you hear the term dehydrogenase, you have to understand that one molecule of NADH will be produced or used. Anything can be there. Here we are talking about production only. So here only NADH will be used, not produced. Produced, not used. Fine. So from isocitrate dehydrogenase, you get alpha ketoglutarate. Clear? Which is also known as alpha Kg or just ketoglutarate. This alpha ketoglutarate is a 5 carbon compound, but isocitrate was a 6 carbon compound. So what happened? During this process also there is oxidative decarboxylation. Again oxidative because the hydrogen and electrons from this isocitrate are removed. Okay, so isocitrate becomes oxidized and one carbon dioxide is removed. So there is decarboxylation. So there is oxidation and there is decarboxylation. So it is called oxidative decarboxylation of isocitrate gives you alpha ketoglutarate. Clear? So along with alpha Kg, you are getting one carbon dioxide and one NaDH. So till now, from the pyruvate, which was 3 carbon compound, we have removed one carbon as carbon dioxide at pyruvate dehydrogenase complex, and second, we removed here an isosocitrate dehydrogenase complex. Clear? Fine. Now this alpha ketoglutarate, it again undergoes oxidative decarboxylation. Clear? So this 5 carbon compound alpha ketoglutarate, it undergoes oxidative decarboxylation. There is one more molecule of NADH produced and one molecule of CO2 removed. This gives you succinyl-CoA. What is not shown in this diagram is that coenzyme A is added again to this reaction. Okay, so there is coenzyme A added to this reaction. So alpha ketoglutarate, okay, will get converted into succinyl-CoA. Again, there is oxidative decarboxylation. You are getting one NADH and one CO2. By this step, you have lost all the 3 carbons from pyruvate. Okay, they have been lost in form of carbon dioxide. Clear? Succinyl-CoA, okay, succinyl coenzyme A is now converted into succinate. Succinyl-CoA, succinate are both 4 carbon compounds. Clear? And this happens with help of an enzyme called succinate synthase, because it is synthesizing what? It is synthesizing succinic acid or succinate. During this process, the coenzyme A is again removed. Okay, so it is getting removed, and one GDP, okay, GDP, guanosine diphosphate, gets converted into GTP, guanosine triphosphate. Now, this GTP is equivalent to ATP. Okay, you can just consider it as ATP. So, at this step, from succinyl-CoA to succinate, you are producing one molecule of ATP. This is again an example of substrate level phosphorylation. We had seen substrate level phosphorylation in case of glycolysis. This is another example of substrate level phosphorylation. Clear? So till here, any problems? We have started with acetyl-CoA, 2 carbon, plus oxaloacetate, 4 carbon, gives you citrate, 6 carbon. Citrate to isocitrate. Isocitrate oxidative decarboxylation gives you one molecule of alpha Kg. Alpha Kg, again oxidative decarboxylation gives you succinyl-CoA. Succinyl-CoA gives you succinate. Fine. Now this succinate, it gets oxidized. Okay, the succinate is again oxidized into fumarate, and this happens with help of an enzyme called succinate dehydrogenase. Remember this enzyme, it is an important enzyme. Why? Two reasons. First thing, we discussed that tricarboxylic acid cycle takes place in the mitochondrial matrix. This is true. All the enzymes are present in mitochondrial matrix except succinate dehydrogenase, which is present on the inner membrane of mitochondria. Clear? Second thing, this is the dehydrogenase which will give you one molecule of FADH2 and not NADH. Okay, it is producing FADH2 instead of NADH. Clear? Flavin adenine dinucleotide. Clear? So this enzyme is important for two reasons. Clear? From succinate, there is dehydrogenase. Okay, so there is oxidation. It will give you fumarate. Fumarate, there is addition of water. Okay, so fumarate is condensed with water to give you malate. Malate also you have heard of in CAM pathway, in C4 pathway, right? So we are back at malate or malic acid. This malate, there is the last dehydrogenase enzyme called malate dehydrogenase. This malate dehydrogenase converts malate into oxaloacetate. So you are back at the beginning of the cycle, and during this process one molecule of NADH is produced. Clear? So during the Krebs cycle, just count. You are releasing 2 carbon dioxide. Okay, so 1 and 2. Let me just show you. Okay, so 1 carbon dioxide and 2 carbon dioxide. Yeah, there are 3 NADH produced. So 3 dehydrogenases which are producing NADH. 1, 2 and 3, and 1 FADH2 is also produced. During this whole process, there is one substrate level phosphorylation which will produce one molecule of GTP. GTP is equivalent of ATP. Sorry. Yeah. So these are the reactions, and this is the basic tricarboxylic acid cycle. Here it is very simple, and I will also show you the tricks to remember all of these points. How can you remember all of them? Clear? From oxaloacetate, as you can see, the cycle begins again. A new acetyl CoA comes and the cycle continues. So this is the whole reaction, the whole cycle which we just saw, along with the diagrams. Okay, so the structures are also shown here. Why are these structures important? Because they show you from which side or what bond is broken, okay, and how the reactions are taking place. Again, these structures are not very much important, okay, for your exam point of view, but they are important to understand. If you are that type of a person who requires structures to understand a process, then this will be helpful for you. Otherwise, this diagram is good because it shows you the number of carbons in each molecule. It shows you the proper the enzyme name and the reactions that are taking place during this um, process, okay, the whole cycle. It is shown in very best way. Fine. So this is the whole reaction of TCA cycle. If you want to write it down, write down 1 pyruvate. Okay, so 1 molecule of pyruvate plus 4 NAD plus. Clear? Why? Because pyruvate, remember, pyruvate has to be converted to acetyl CoA, during which one more NAD plus is produced. So 3 NAD plus from TCA cycle and 1 NAD plus from pyruvate dehydrogenase complex. So 1 pyruvate plus 4 NAD plus 1 FAD plus for succinate dehydrogenase. 1 molecule of water and 1 GDP plus inorganic phosphate for the succinyl CoA to succinate transition, right? This whole reaction will give you 3 molecules of carbon dioxide which come from pyruvate, 4 molecules of NADH plus 4H plus 1 molecule of FADH2 and 1 molecule of GTP. So this is the whole reaction. Now how to remember the TCA intermediates? Okay, how will you remember all of these molecules that we just studied? Remember this mnemonic, okay? Can I keep selling soda for money, officer? Okay, so there is this little kid that you can see and he's selling soda. And he's asking the police officer if he can keep selling the soda for money. Okay, so this is the question he asked. He asked, can I keep selling soda for money, officer? He's asking, can I keep selling soda for the money? So if you see, this is the names. Okay, how will you remember? So can, C, is for citrate. Okay, the first molecule formed is citrate. So can means for citrate. Then isosocitrate. So can I. K is for ketoglutarate. Okay, so keep means ketoglutarate. Selling, succinyl CoA. Soda, succinate. For fumarate. Money, malate. And officer, oxalate. Can I keep selling soda for money, officer? Citrate, isocitrate, alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, and oxaloacetate. Now for people who may not be able to remember the same way, we can like the enzymes. If you cannot remember the enzymes directly, for them this is the mnemonic to remember the TCA cycle enzymes. Okay, so there are these two kids, okay, and they are drawing some paintings, and there is a doctor, so they are painting the some things or drawing something for the doctor. So what is the statement for this? It is Shruti and Dhara drew 7 drawings for doctor. Okay, so Shruti and Dhara, they drew 7 drawings for doctor. Shruti and Dhara drew 7 drawings for doctor. All of the D's here are dehydrogenases. Okay, the only thing you have to remember is before the dehydrogenase you have to put the name of the substrate, okay, or of the product. Let us see one by one. So Shruti, the first one, is actually citrate synthase. Okay, you just have to put the name of the product, which is citrate. What are we synthesizing? We are synthesizing citrate in the first reaction, right? Here citrate is synthesized, so citrate synthase. S for synthase. A is for aconitase. From citrate to isocitrate is enzyme called aconitase. So Shruti and Dhara again is for dehydrogenase. You just have to put the name of the substrate. Okay, so isocitrate dehydrogenase. Then comes alpha ketoglutarate dehydrogenase. Okay, so it is again a D. Dhara Dru. So dehydrogenase. You just have to put the name of the substrate, which is ketoglutarate. Then 7 here is for succinyl CoA synthase. Again, like citrate synthase, succinyl CoA synthase, you just have to remember S means synthase. Clear? So Shruti and Dhara. So synthase, aconitase, dehydrogenase, synthase, dehydrogenase, fumarase, and again dehydrogenase. That is the path. Okay, that is the order. Again, after succinyl CoA synthase comes succinate dehydrogenase, which is the special enzyme I told you to remember. After which comes fumarase, and after fumarase comes malate dehydrogenase, and this is how you have to remember the name of the enzymes. Shruti and Dhara drew 7 drawings for doctor. So this is the summary. Okay, you are in the input is acetyl CoA plus 1 ADP and inorganic phosphate, 3 NAD plus and 1 FAD. The product is 2 molecules of carbon dioxide, 1 molecule of ATP, 3 molecules of NADH from NAD plus and 1 molecule of FADH2. So this is the output from tricarboxylic acid cycle. So in the next class, we shall be beginning with the electron transport chain, okay, in the next processes. So see you next time.